Good afternoon, folks. My iPhone tells me it's 3 p.m. sharp, so since we got the keeners here, and I'm sure you want to go and enjoy what nightlife Atlanta has to offer afterwards, I'm going to be the ones between that and your evening. So I'd like to thank Bernie for the invitation to come to speak today at this regenerative medicine meeting. And I guess my talk is going to be of a slightly different tone. A lot of people are cheerleaders for the field, and the glass is consistently half full. I'm going to be looking at it from the perspective the glass is maybe half empty, but with a smile on my face, because I think there are remedies for the field to move forward. So that being said, I'm preaching to the apostles here. Everybody's a believer in cell therapy. A number of clinical trials. Now, these are not clinical trials of meat and potato bone marrow transplant. I'm talking about experimental cell therapies, and this is from a terrific database that Alexei curates on his website, and he hasn't updated the 2015 data, but I invite you to take a look at that website. Now, the U.S. leads the field. China is very, very enamored with the cell therapy platforms, and you have a whole bunch of other players. Again, this is data that Alexei curated and using what's published in clinicaltrials.gov. And the king of the field are mesenchymal stromal cells. By far, these are the most popular non-traditional cells in clinical use, really still in exponential growth. T cells are very hot, of course, especially with the advent of CAR T cells. Everybody wants to cook some CAR T cells in their garage to give to somebody. And of course, there's the clutch of all the other cells that are playing down here. So MSCs, I think, if we're going to be looking at a cell product that's out there, that's fairly mature, that could inform us of a pathway to follow, that's the platform we should be examining. Now, for those who are not familiar with the mesenchymal stromal cells, there's really two buckets of stem cells in your bone marrow. The hematopoietic stem cells, which give rise, they're CD34, sort of a crude marker, give rise to all the blood products. This is the first and last time I'm going to be talking about hematopoietic stem cells. And then you have mesenchymal cells. Now, mesenchymal cells, you got to understand, are very rare nucleated cells in your bone marrow. 1 out of 100,000 to maybe 1 out of 500,000 is a mesenchymal stromal cell. So the notion that you can yank out a gallon of marrow and take the MSCs out of that, use that up front, is really not practical. Now, Arnie Kaplan coined the term mesenchymal stem cell in the early 90s because when you take these cells and you put them in culture in a Petri dish, very simply they stick to plastic, the hematopoietic cells float off, and then these start proliferating like fibroblasts. And they just go and go and go and go and go and go without you having to do anything. So in the era prior to synthetic cytokines, it was very easy to grow mesenchymal stromal cells in vitro. And what was discovered is if you add either steroids or you add TGF-beta or vitamin C, you can tweak them into differentiation into bone, fat, or chondrocyte precursors, hence the mesenchymal progenitor concept. And early on in the field, a quarter century ago, everybody thought this would be the cat's meow to do bone repair. So it was all like bone repair stuff. But what has been discovered since is that there are MSC-like cells all over your body. Now, people get their knickers in knots about MSCs are stem cells, they're not stem cells, and the MSCs from fat are different from bone or different from teeth. I think they're all 6 degrees of separation. I lump them together as MSC-like cells, and the bulk of the MSC-like cells outside of your bone marrow are almost certainly pericytes. Now, pericytes are cells that populate, basically, you have your capillaries, your arteries, arterioles, your capillaries, and you have the postcapillary venules. And the capillary and the postcapillary venules is where you have these pericytes, and you also have macrophages. And these pericytes can also be harvested, and that, stromal vascular fraction from fat, and you put that in culture, it's all pericytes. People say they get MSCs from liver or brain or whatever. It's all parasites. And they very much behave like MSCs when you culture expand them in vitro. So they're prevalent. And what also has been, we've learned over the years, is initially people thought that you could squirt in MSCs into tissue and they would trans-differentiate into that tissue and do tissue replacement. All of that is bunk. They don't do that. What MSCs do is they secrete factors. And again, Arnie Kaplan likes to coin that term. And the factors fall basically into two buckets: anti-inflammatory and tissue repair. So, isolation of MSCs. I'm a hematologist. I'm a clinical hematologist. You know, you have those dimples in the back right here? So I go in, I put a little bit of freezing like the dentist does, and I go in with a trocar under local anesthesia. In my office, I could pull out 20 cc's of marrow, pull out a trocar, put a Band-Aid, you can walk out of the office and go play golf. It's as easy as that. You take that 20 cc's of marrow and you put in the Petri dish I spoke of, and having just 10% fetal calf serum and DMEM or alpha or whatever you want, the MSCs stick to plastic, they start growing, and they grow and grow. They have a phenotype, which is a clutch of cell surface markers, which are non-specific. A lot of mesenchymal tissues express these, 105, 73, 44. Probably, if the most precise reductionist cell surface marker, I would suggest is CD271, which is a low affinity nerve growth factor, which probably enriches the most the clonogenic precursors. What's important is they don't express CD45, which is a pan-hematopoietic marker. Again, when you grow cells in vitro, at first monocytes will stick to the plastic. You don't want to mix up monocytes and MSCs. And they don't express PCAM1, which is an endothelial cell marker. And again, the Petri dish, that's what they look like. Mouse, human. Actually, mouse are the most difficult MSCs to grow. MSCs from humans, monkeys, pigs, dogs, horses, they grow very easily. So I described the procedure. And you're going to ask me, why is it MSCs really are a solution looking for a problem to fix? Is because one property and one property only, and it's this one, is if I take 20 cc's of your bone marrow and I put it in culture, within 2 to 3 weeks, I can generate a gram of your cells, 10 to the 9 cells. And if I know relatively what I'm doing, and especially if you use platelet lysate, I can generate a kilo of your cells. So these cells, even in mature adults, have a really robust proliferative capacity in vitro, as opposed to, say, skin fibroblasts that'll poop out after a week and they'll become senescent very quickly. So you grow these and you can have a cell pharmaceutical. So here's a perfect solution looking for a problem to fix. Now, the two types of problems that are fixed are regenerative medicine and immune modulation. I'm going to focus on immune modulation. Now, the first piece of data, you see, MSC is a fascinating field because the first clinical trial done in people was done without any animal data. It was done just on the in vitro basis. The first real meaningful animal data was actually published by here in a specialty journal. It was sponsored by Osiris, Yonder. And what they did was basically, this demonstrating immune modulation. A monkey, they took skin from monkey A and put it in the forearm of monkey B. And skin is the most immunogenic tissue you could possibly transplant across the HLA barrier. And if you do that, after 7 days, that piece of skin will fall off. If you gave MSCs to the monkey, it took 11 days to fall off. So this was the first piece of data that MSCs are really immunomodulatory in vivo in a simian model. Helen launched 1,000 ships. The paper that launched dozens of clinical trials is this one, looking at MSCs for graft versus host disease. So Katarina LeBlanc is an MD PhD bone marrow transplanter at the Karolinska Institute in Sweden. Here we go, there she is. And she published a case report in The Lancet. In modern medicine, to publish case reports in high-impact journals is virtually impossible. That's really old school. But the reason she got away with it is the following. This 9-year-old boy was dying of graft versus host disease after an allogeneic bone marrow transplant. So if I take OHAD's marrow and I put it in you, and there's a level of mismatch, the T cells from OHAD's donor marrow will attack your liver, your skin, your gut. That's called graft versus host disease. 30% of patients get that, and a goodly fraction will die from the gut involvement in particular. So here, this young boy had an allotransplant, and the open circles are stools, which are a surrogate of diarrhea, and the black circles are bilirubin of liver dysfunction. And what Katarina did, and in Sweden they can do this because they have this dispensation, so they can do crazy stuff. If somebody is like dying and there's no option, and they can just say, I'm going to do this because I think it's going to work. So she went to the boy's mom, collected mom's marrow, grew mom's MSCs, that's haploidentical to be technical, and gave the boy mom's MSCs at a dose of 2 million cells per kilo. Here, there was a response, there was a relapse in GVH. She gave a second dose, you see how many days after transplant. The boy went to complete remission of his graft versus host disease. Now, eventually, his leukemia relapsed and he died. But this was so remarkable because when you have grade 4 GVH, it's a death sentence. And this was truly a Lazarus effect. So this was followed by a series of academic-sponsored phase 2 studies, the largest of which, again, Katarina here, in Lancet, 55 patients, a large multinational grouping in Europe, and we're getting terrific response rates. 70% of patients were getting either a complete or a partial response to the graft versus host disease. Truly astonishing. Multiple phase 2 studies were replicating that data. The field was on fire. So the logical next step is, it's completely logical, industrialize it. So the data that I showed you initially was IP that came out of Case Western, which eventually made its way to Osiris. And there the idea was they would take a bone marrow from a young college kid, I presume, give him $200, I presume, and grow from their website, this is from their website, 10,000 doses from each donor. And they would freeze this off, and they could ship those to the East Dakota Cancer Center, and all they had to do there is take the frozen cells, thaw them the way you would thaw fresh frozen plasma, hang it, and give it to the patient, and they would treat all sorts of the ailments. I'm sorry to see that they're not treating male pattern baldness, but that's a different discussion. So, completely logical. However, in 2009, they issued a press release. Now, this is now 6 years ago, about the prospective phase 3 randomized study where they failed to meet their primary endpoint of getting complete remission of graft versus host disease in steroid refractory patients. So in their hands, MSCs was no better than a sugar pill. They've never published a paper. They've presented this as an abstract at a meeting. That's it. So I can't comment on the rest of the data and how it was analyzed because I haven't published it. So this is a bit of a bummer. Now, Athersys, I love Athersys because I love Bob Deans, who's moved on, but anyway, they have another MSC-like product. They'll say, oh, it's different, it's MAPCs, it's a technology from Catherine Verfa. It's an MSC-like cell, universal donor. They claim they can grow 1 million doses from a single donor. In April of last year, they tested their product in subjects with ulcerative colitis, which is an autoimmune disease of large bowel, and it didn't work. They did not meet their primary endpoint of efficacy. April was not a good month for this company, because in April of 2015, this year, they tested their product in adult subjects with acute stroke. And they were giving it IV in stroke, and they were asking would it improve the outcome of stroke, and the answer it did not. They did not meet their primary endpoint of efficacy in stroke. So here we are. I haven't shown you a single piece of mouse data. Now, I did a PubMed this morning, because I like to do so before talks. Mesenchymal stem cells in mouse, you'll get 9,753 hits. I'm not making up this number. I have published dozens of papers demonstrating that MSCs in murine models will have not only a statistically significant but substantial effect on a wide array of immune inflammatory disorders. This has been replicated ad nauseum in multiple labs across the world, academic and industrial. So you have this overwhelming weight of preclinical animal data that says this should work. You have phase 2 academic trials done in Europe predominantly showing a strong signal, yet the industrial studies failed. Now, I'm Canadian. I moved to the U.S. 6 years ago. I was at McGill. I had to come to the U.S. My grants didn't come with me. Boo-hoo. I had to write a whole bunch of NIH grants. So you write NIH grants, and, so I'm an MSC expert. Well, at least I think I am. So I'm an MSC expert. And, I get this pig sheets, and, that's like getting a double open-handed slap across the face. And you say, Galipeau, you schmuck. All these industrial studies are negative, move along, do something else. This is a waste of your time. So I sat in front of a piece of paper with my pencil, and I was figuring, what is it that I can say to these skeptical, quarrelsome, stiff-necked NIH reviewers that would possibly convince them that MSCs aren't dead, that there's actually a rational explanation that can be hypothesis-driven and remedied, understanding why the industrial studies failed, yet the academic and preclinical animal data works. And here are 4 points I'm going to bring up for discussion today. First, donor variance. I told you, FRCS, 1 donor, 1 million doses, and I told you that Osiris, 1 donor, 10,000 doses. So let's talk about a bit of cell biology. MSCs, this is a cartoon from a review written by Wim Fibby a number of years ago. You see a gazillion pathways MSCs work. When you see that, immediately you're skeptical because that means that nobody knows how it really works. But the one thing you need to remember is this. Interferon gamma stimulation of MSCs in vitro substantially deploys their immune modulatory properties. They become profoundly immune suppressive when they're interferon gamma activated. Therein lies probably the Rosetta Stone to understand how they tick. So there are certain well-understood mechanisms of action by which well-documented suppressor cells like Tregs and dendritic cells can attenuate the immune response. And that's production of IL-10, TGF-beta, COX-2, which makes prostaglandin, and IDO1. And what we did is we took MSCs from normal human donors before and after interferon gamma stimulation and looked at the RNA of these genes, and we found that MSCs never, ever, ever make IL-10 under any circumstance, ever. They make a bit of TGF-beta, but it's not upregulated by interferon gamma. But both COX-2 and IDO1 are massively upregulated at the RNA level. Now, IDO1, we went on to show by Western blot, you need to do that, the protein actually goes up as well. It's virtually absent in culture-expanded resting MSCs, which is the stuff the companies inject, but interferon gamma activated IDO goes up. So how do we test functionally this activity? So we do what's called a T-cell suppression assay. I'll summarize how the assay's done. I'll take 10 cc's of your blood, and then I put it in a centrifuge, and then it separates, if you put a bit of Ficol, you get the red cells and the neutrophils in the bottom, and you get at the interface lymphocytes and monocytes. That's called the Buffy coat. And if you look at the Buffy coat by flow cytometry, what you have here just by side scatter, forward scatter, are about monocytes and lymphocytes, about 4 to 1. So a Buffy coat is a gamish. This is what you have in unfractionated blood, all the granulocytes, those are removed. So this is what we use, and what we do is we take these T cells and we add the Dynabeads, which is something you buy commercially, has anti-CD3, anti-CD28, cross-links a TCR and a co-stimulatory molecule, and it fools the T cells into start proliferating, sort of mimicking immune activation. And if you load the cells with a dye called CFSC, and they're not stimulated, they're high, but if they're stimulated, as this daughter cells split 2, 4, 8, 16, so on and so forth, it dilutes the dye, and you get this big comet tail here. So we looked at the MSCs of a whole bunch of normal human donors, number 303 to 311, and if you add one MSC per 3 T cells activated, it completely blocks T cell proliferation. You see, this is proliferating T cells, blocked. And if you dilute out the MSCs to 1 for 27 T cell, the T cells sort of overcome the MSC effect, they start growing. But if you look at this middling ratio, 1 to 9, you'll notice that donor 308 is still very suppressive, yet the donor 305 is much less so, which suggests that your MSCs may be more suppressive than her MSCs. Absolutely. So there you go. Why is that so? So what we did is we took 4 normal volunteer donors, grew their MSCs. There's no IDO protein in resting cells. We stimulated them with interferon gamma. If you add a bit of TNF alpha, it jacks up the effect. And the IDO protein goes up. If you normalize to a loading control and you do a ratio, what you find is you can normalize the amount of IDO. And what we found is, which was not surprising, is the more IDO there is, the more T cell proliferative you are. There's a good correlation. So why does this matter? That means that if you're a very good IDO inducer, and the man besides you is a very poor IDO inducer, yet Osiris used his cells to grow 10,000 doses, they're in much more trouble than if they had used your cells. So that's where donor selection and bias may have played a role in industrial studies. Please remember, the academic clinical trials, the academic centers, when they grow MSCs from a donor, they grow at most 10 doses, 10 out of a donor. The Osiris product, they grow 10,000 doses out of a donor. So hang on to that notion there. So the other thing also that we found that if you activate the MSCs with interferon gamma beforehand, I told you they upregulate IDO, and they completely block the ability of T cells to make interferon gamma. And not only interferon gamma, they'll block the T cells from making IL-2 as well and TNF alpha. And you see activated T cells, MSCs work, but interferon gamma work even best. And this is just a different way of activating the cells, again showing that interferon gamma activation augments their immune suppression. And it's believed that if you inject MSCs in a person and they see interferon gamma because their person's inflamed, that's how the MSCs get really jazzed up and start blocking T cell proliferation, amongst others. And again, these assays have been, I'm part of the ISCT, the MSC committee. So these concepts have been enshrined in a couple of position papers of how to interrogate the immune plasticity of MSCs. So how does this work? So here's RNA-seq. I really like this readout because each time somebody shows me, one of my grad students walk up to me, Dr. Galipeau, I want to do a gene chip and do 50,000 genes. I said, I don't want to hear about that. Each time I see 500 genes go up and 300 genes go down, I have no idea what it means. RNA-seq is much simpler because you choose your target genes and it quantitatively looks at RNA levels. So what we did is we took MSCs from a normal subject and we interrogated 47 genes that we chose because in the published literature, people have claimed that these genes listed on the left play some sort of a role in immune modulation by MSCs. So we looked at these cells, and after they had been exposed to activated T cells in vitro, which would be the physiological source of interferon gamma, and what happened to the gene expression. And you'll see here, some of the genes go up in expression quantitatively luxuriously. IDO1, you see blue is low and red is high. So for your convenience, I boxed here those genes that really get jacked up after interferon gamma activation of MSCs. And again, IDO is a very important immune blocking agent, but there are a lot of genes here that are important for regeneration. HGF is important for regeneration. IL-6 is there, VEGF is there. You'll notice PD-L1 goes up as well as an immune blocker. So what we did to be even more reductionist, rather than using activated T cells, we now took MSCs from normal volunteers, a whole bunch of them, and my little thing is going to appear there here. before and after interferon gamma activation. Now they were just added recombinant interferon gamma now. So it's just interferon gamma. And what we found was, again, we had massive upregulation of these genes listed up here on the left. And I'm showing this because this has been published just now. And there are other genes that were published in the literature that people claimed were important in their effect. But in our hands, we actually found the gene was downregulated by interferon gamma. But still, you have a goodly list here of reasonable effector pathways that are involved in immune modulation, regeneration, are upregulated by interferon gamma. And this has real effects on activated T cells. When you co-culture MSCs and activated PBMCs, PBMCs, for example, make a lot of IL-13, but when they sort of tango with MSCs, the IL-13 goes down. Same thing for IL-17. So I'm showing you here, all these cytokines made by T cells go down significantly when they tango with MSCs in a Petri dish, and they're listed here. And here are other genes that are actually proteins that are produced by MSCs that when they see activated T cells get upregulated. So again, there's this crosstalk. So I told you that when we do these assays with PBMCs, peripheral blood mononuclear cells, purified from a phycol, you don't have pure T cells. It's T cells and monocytes together. And people sort of kind of poo-poo the monocytes. So they're just sort of like, they're like watching the show. They're just sort of sitting there watching the show. So we asked the question, do monocytes play a role in the apparent immune suppression of MSCs in vitro? So what we did is we actually removed the monocytes from the PBMCs and did our suppression assay anew. And what we found was that if you add MSCs to PBMCs where it's lymphocytes and monocytes, you have suppression. But if you remove the monocytes, the suppressive effect is markedly attenuated. This suggests that the monocytes are not non-specific bystanders. They play a direct role in MSC-mediated suppression in vitro. And indeed, what we went on to show was that MSCs directly talk to monocytes, and the monocytes develop a M2 macrophage phenotype, and the monocytes themselves start producing a lot of IL-10. Now, this is really important because this is work published by Eva Meze's group at the NIH. And she had this terrific model of sepsis. She takes mice, she slices open their belly, she ties up their colon, and she sticks a needle through it. She sews up the mouse. The bacteria from the colon seep into their blood. They get blood poisoning, sepsis. The mice die. If she gives the mice MSCs IV, the mice don't die. Really clean readout. And this is down here, the survival curve. Septic mice with MSCs, septic mice without MSCs. But if, a priori, she depleted the mice of monocytes before giving the MSCs, the therapeutic effect was completely lost. And what she showed was this imaging on the left. I'll walk you through it. When you inject MSCs in the tail vein of a mouse, it's the same thing as injecting MSCs in antecubital vein in a human. The first capillary bed encountered will be the lung capillaries. The MSCs microembolize in the lung. Everybody knows that. 99.99% of the cells go to the lung. They stick around for 1 to 3 days, then they go away. A minuscule fraction may redistribute at sites of inflammation, but the bulk of the cells are in the lung. And what she found was that the MSCs egress out where the capillaries and postcapillary venues are and start interacting with the monocytes. And about half of them sort of kissing the monocytes, and the other half, the monocytes actually phagocytose the MSCs. So she went on to show that this MSC monocyte crosstalk was really important because if you remove the monocytes, it doesn't work. If you use a mouse that's IL-10 knockout where their monocytes can't make IL-10, it doesn't work. And she showed that the donor MSCs, if they express COX-2, was important because if they were COX-2 knockout and prostaglandin, it didn't work either. So she came up with this scheme. I like this model. It's all mouse now, no human. That MSCs will upregulate COX-2 if they're stimulated with LPS or interferon gamma. COX-2 makes prostaglandin, which then activates the monocytes, and the monocytes make IL-10, and that's how she comes up with this model, which makes a lot of sense, that she's in essence suggesting that MSCs convert the lung to an immune endocrine gland, making IL-10. So this cartoon we published, it summarizes 10 years worth of work. You know, I know it brings tears to your eyes that 10 years brought this, but interferon gamma will upregulate IDO1 in human. Mouse doesn't do it, which is vitally important in vitro. It converts tryptophan to kynurenine, which can block T cells and monocytes. And we have shown that IDO plays a role in converting monocytes to IL-10 producing M2. We've recently published that interferon gamma activated MSCs also upregulate PDL1, which also blocks T cells. And I would propose the following model that MSCs produce COX-2. Actually, COX-2, they don't produce COX-2. COX-2 makes prostaglandin, IL-6 and HGF, which can directly talk to monocytes and also lead to increase in IL-10 production. So this whole thing that nobody knows that MSCs, how they work, is bunk. We know exactly how they're going to work. We have a very good sense where we're going here. So, the other big thing, in the early days of the MSC field, at least the industrial narrative was MSCs are magical. They're just magical because I can take your MSCs and I could give them to everybody in the room and nobody will immune reject them. They're immune privileged. There's something special about them, especially if they're fetal. If they're fetal MSCs, you can give them across species. They're immune privileged. Is that true? Because that may play a role in why they don't work, because when Osiris made 10,000 doses and they're giving 2 doses a week for 4 weeks in a row, are people developing an immune reaction against that? If you're going to treat Crohn's disease, you're not going to cure anybody by giving a couple of doses. It's going to be like Remicade or anything else. It's going to be once a month for the rest of your life. Will you get immune sensitized? So, is it possible for MSCs to get to even be immunogenic? Well, look, if you do flow cytometry, MSCs, like any somatic tissue, express MHC class 1, and it'll be upregulated by interferon gamma. Resting MSCs do not make MHC class 2, but if you stimulate with interferon gamma, they make MHC class 2, class 1, class 2. You're telling me they're immune privileged? They make MHC class 1, class 2, which is what antigen-presenting cells do. They will not upregulate co-stimulatory molecules, CD80, CD86, but will upregulate PDL1. I already showed that. So to test the hypothesis, are they immune privileged, we did a simple experiment. Black 6 mice and bulb C mice are completely MHC mismatched. Completely MHC mismatched. And we're going to use a black to white icon to make this simple for you. So to track these cells in vivo, we used a gene therapy trick. We grew MSCs from a black mouse and gene engineered them to make erythropoietin. MSCs don't make EPO, but you know EPO, that's a drug that Lance Armstrong, shot up so his blood counts go up. Well, when you put EPO producing MSCs in a mouse, you can track if the MSCs are still alive by measuring the hematocrit over time, because if the MSCs go away, the blood counts will go down. So what we did was, here, if you take MSCs from a black 6 mouse have been genetically engineered to make EPO and put them in a black 6 recipient, the syngeneic, the same as autologous, as your own. The hematocrit of the mice, the red cell count, goes from normal to way high and stays there for a long, long, long time. Mice can tolerate a high hematocrit, they won't get a stroke. If you do use the exact same black 6 MSCs making EPO and put them in a completely mismatched bulb C mouse, you get a spurt of increase in red cell, but it drops down over time, back to baseline. So we figured, oh, well, maybe we messed up. Let's give another dose. So we gave another dose. What happened is, again, this is spaghetti plots, or this is each individual mice. The increase was less, and the duration was shortened. And we gave a third squirt, hardly any effect at all. This is a classic pattern of alloimmunization. MSCs are not immune privileged. Don't let anybody convince you about that. Now, and orthodox transfusion medicine will inform that. When you get transfusions, you get either red cells, which don't express MHC molecules, or platelets. Platelets express MHC class 1. And it's well known that if you receive random donor ABO matched platelets, that about 40% of people eventually get alloimmunized within 28 days. So it's well described that when you infuse in a person IV MHC class 1 mismatched cells, a goodly fraction will get alloimmunized. Now, other groups have now replicated the data we've published. Vimfibi showed that. Actually, Don Finney went a step further because he actually injected MSCs in the brains of monkeys. Brains are supposed to be immune privileged, like your eyeball type of thing. Even though he did that, the monkeys developed an immune reaction against mismatched MSCs put in their brains. So repeated transfusion of unmatched MSCs may lead to an alloimmunization. I didn't say will lead, may lead. Culture expansion. Now I'm going to dwell upon this because I told you the industrial studies didn't work. Now I talked about donor variants. We don't know how they chose their subjects or if they used the right parameter to select the donors. I just showed you that allo cells can get immune rejected. How about culture expansion? So the 5-dose versus 10,000 versus a 1,000,000 dose paradigm. And I'm not making these numbers up because 5 doses is what academic centers do. 10,000 is Cyrus, 1,000,000 is Athersys. So when you do in vitro expansion, people talk about passages and this and that. Let's just say that most typical clinical doses are grown up within about 2 to 4 weeks to generate a couple of doses out of a donor. And anything beyond that is whatever you want. So the question we asked was, are replication fit MSCs equivalent to senescent MSCs? So senescence is a phenomena that it's called Hayflick's limit. Hayflick's a guy in the 60s that took cells from fetuses and asked the question, how many times will these double? And the limit is sort of kind of 52 doublings. Now, true exponential 52 is a very, very large number, but that's sort of kind of the limit. Unless the cells become transformed, and then you have HeLa cells, and that's a different kettle of fish. So you take MSCs, and at day 0, you split them, they grow, you reseed them at low density, they grow, you reseed them at low density, they grow a bit. And so as you go on, eventually after, say, 40 days in serum, that's the only way we can generate senescent cells is using serum. It's much harder actually if you use platelet lysate, but that's a different discussion. They stop dividing. And there's markers for that senescence. The cells are beta-gal positive. If you do flow cytometry, they're big fat cells. Who here works with MSCs? Anybody works with MSCs? So, you know the big fat round cells, those are senescent cells. The little tiny spindly ones, those are replication fit. And they're all in G0. So G0, big fat, beta-gal positive. And they're virtually indistinguishable by phenotype with fit cells. So CD90, 44, all the ISCT markers are identical. But they can't suppress T cells in vitro. So this is a slightly more sophisticated readout for T cell proliferation. It's CFSC dilution in interferon gamma. You beat activate T cells, they proliferate, they make interferon gamma. You add fit MSCs, they block all this. You add senescent MSCs. Senescent MSCs actually stimulate T cell proliferation. And this is just enshrined here. So senescent MSCs have an intact phenotype. If you grow your cells because you want to generate the gazillion doses, you won't be able to tell them apart unless you interrogate for it directly. They have an altered transcriptome. I didn't show that data. Immune suppressive properties are messed up. And there's some clinical data that suggests that late passage MSCs are not as good. Again, this is out of Sweden, Ole Ringden and Katarina LeBlanc. They've treated so many patients for GVH, and they had sort of late passage production runs and early passage production runs. And again, this is retrospective, but patients that received MSCs that were more late passage production runs, their outcome was not as good as early passage. So again, it's soft retrospective data that suggests that making 5 doses out of a donor is probably better than making 10,000 doses out of a donor. Okay. So, if there's one thing I want you to remember at the end of this conference on a Saturday afternoon is this. MSCs are like sushi. So, who here will go buy sushi for their partner and say, you're flying back tomorrow from Chicago, okay, I'll put the sushi in the freezer for you, and tomorrow you can thaw it and eat it. Nobody would think about that. Any sane graduate student whose PhDs depend upon murine experiments for them to graduate using MSCs injected in the animal, never, ever use frozen cells. I mean, they could take cells out of the freezer, put them back in culture, make sure the cells are growing and they're perfect. And I don't know about the students in your lab, the students in my lab, they read books to their cells. They come in on Sunday to change the media, to make sure they're perfect. They harvest the cells fresh. They put them on ice. They run to the animal facility. They inject the cells into the mice. 99.9% of the published preclinical data in animals use fresh cells. So, we went from, I told you, 9,753 papers, mesenchymal stem cells in mouse, virtually all of which use fresh cells, to clinical trials where what do? We produce these cells, massive expansion, almost certainly all senescent. You harvest, you freeze them. And then how it's done is they thaw the bag at the bedside, and the thawed cells are delivered IV to the subject within 4 hours. So the question we asked was, are thawed cells the same as live cells? So if you take cells straight out of the freezer, there's a readout for dead and dying cells. It's PE and exin, and MSCs, human MSCs, you'll have a lot of dead cells and dying cells. You have live cells. And that's okay, it's kosher. The FDA tells us that you can inject a product in humans as long as no more than 30% of the cells are dead. And that's okay. Now, these are cells that are kept in culture continuously. Of course, they're way more live cells. And exin PI is very much more sensitive than trypan blue because this is indistinguishable, by the way. So the question we asked was, are thawed cells as good as live cells? These are the exact same MSCs. And of course, you anticipate the answer is, of course not. So here you have T cells that are proliferating, and if I add fresh, live, fit, perfect, happy MSCs, they completely suppress T cell proliferation. If you take cells straight out of the freezer and put them in the Petri dish to activate T cells, they work no better than spit. Now, you can do a more sophisticated experiment where you do a dilution of MSC to PBMC ratio, live versus thawed cells, and the live cells are always superior in suppression. The cryo will catch up. So they're not totally dead, but they're clearly blunted in their ability to at least block T cell proliferation in vitro. And what we found was, you took the thawed cells straight out of the freezer. This is MSC proliferation in vitro, a control. This is T cell proliferation in vitro, or a control. If you add thawed MSCs, I told you they don't work. No. But if you take the exact same thawed MSCs and put them back in culture for a day, for them to sort of recover emotionally from the thaw, you know, like the movie Aliens, you know, they come out of cryo-freeze and they're coughing and all that, huh? They completely recover their immune suppressive properties within 24 hours. So the freezing, there's nothing wrong with the freezing. It's all about the thawing. And we repeated this with multiple donors. Why? Why is that so? So we basically use our interferon gamma trick. So if you take cells straight out of the freezer and you stimulate them with interferon gamma and you look for a readout, upregulation of IDO protein, thawed cells immediately post-thaw can't upregulate IDO. Whereas if they put them back in culture for a day or a week, they can. Why? A bit of cell biochemistry. Interferon gamma binds to its receptor. Thawed cells still have the interferon gamma receptor. And then there's phosphorylation of STAT1, which then leads to DNA and upregulation of IDO and the Gamish stuff. So I showed now what we found was that thawed cells have a blunted phospho-STAT1 response. So basically, the machinery downstream of the switch doesn't work no more. You put them back in culture, it recovers. Why? Because thawed cells undergo a heat shock response. So here we looked at heat shock proteins in 3 different volunteer donors, and we did a time course, and you see heat shock protein 70, A, and B are massively upregulated immediately post-thaw, because thawing is not a physiological thing. So it's really acute cell injury. And it reverses, and as soon as the heat shock protein goes back to normal, the cells start proliferating again. So basically, heat shock proteins are to metabolism what P53 is to DNA. And what about homing in vivo? So to ask that question, and again, none of these in vitro tests foreshadow this. We took human MSCs from men, injected them in the tail vein of black 6 mice, and we can interrogate. I told you, we inject the tail vein, all the MSCs go to the lung. So we fished out human MSCs in their lung by killing the mice a day later, taking out their lungs with PCR for human DNA. So if you inject live cells in a mouse, 24 hours later you kill the mouse, it's very easy to detect DNA. This is the number of PCR cycles on a PCR machine. The more cycles you need, the less DNA there is. So here, there's lots of human DNA in the lung, and it disappears after 48 hours, and we can't find it anywhere else. If you inject the exact same MSCs from the exact same donor right after thawing, we actually cannot detect any human DNA in the lungs of the mice at 24 hours. It's the same as if we had injected salt water. And we looked at all the other tissue in the mice. The spleen, the bone marrow, the kidneys, nothing. The cells are cleared like that. Why? One explanation is the following. So when you freeze cells, the cells undergo a process that's called vitrification. It's descriptive. The cytoplasm becomes like glass. That will actually completely depolymerize your actin cytoskeleton. That was described a quarter century ago. So here, this is phalloidin staining of live MSCs. You trypsinize, you just reset them down, and they have very beautiful cytoskeletons. If you take cells straight out of the freezer and you let them sit on plastic, their cytoskeleton is all messed up. And it's actually reduced in quantity. So basically, when cells are thawed, their actin cytoskeleton is depolymerized. So basically, it's a bag of broken bones. Does that impact on their biodistribution? So what we did is we took live cells and treated them with cytokinesin D, which is a F-actin depolymerizer. It doesn't kill the cell. And when we did biodistribution studies with these cells, this completely replicated. So cytokinesin D cells were not, human cells were not recoverable in mice. So these were live. They were not heat shocked. So there's a plurality of things that together affect thaw cells. Now, Katerina LeBlanc's group showed another phenomenon that's actually fascinating. If you put MSCs in the bottom of a Petri dish and you had whole blood on top, MSCs will initiate coagulation. MSCs express tissue factor. Anyway, if they're hematology geeks here, they make tissue factor, put blood, initiate coagulation. If you do the same thing with thawed MSCs, the clot formation is way more vigorous. Thawed MSCs initiate clotting. She also showed that thawed MSCs in vitro are susceptible to complement-mediated lysis. Live cells, much less so. The thing we found was that if you take MSCs live and thawed and you add activated T cells, and activated T cells with dynabeads, the cells start making perforin and granzyme. They just go crazy. And they sort of lose their stuff in the media. Live cells are quite tolerant to activated T cells. Thawed cells get lysed by activated T cells in vitro. And for example, here, live cells, 1 MSC per 2 T cells, we still can recover a lot of MSCs. You use the same ratio in thawed cells, much reduced. Here, 1 MSC per 4 activated T cells, lots of MSCs, all the cryocells are gone. So, there's nothing wrong with freezing. Nothing. It's all about the thawing. Now, I think the concepts I'm sharing with you, I'm focused on MSCs. These concepts, I believe, are true of any somatic cell that's being utilized in the cell therapy approach. If you have a small company, and all your IP is based on, I'm going to make this up, hepatic cells to treat Parkinson's disease. I'm making this up. And all the data in mice was using live cells. But then the MBA types in your company said, live cells, forget that. That's not deployable. Let's freeze them. And we'll make our business model around frozen cell. But you haven't convinced yourself, you haven't shown to yourself that the frozen product behaves as the same as a live, especially if you're going to thaw it. Beware. I invite you to verify that because you can go down a road that would be very disappointing. So, thawed cells. They undergo a heat shock response. They're susceptible to T cell killing. They're susceptible to complement-mediated lysis. They can initiate coagulation. They have abnormal membranes. That's why I think they're cleared also in vivo in an accelerated way. They have an abnormal cytoskeleton. But other than that, they're perfectly fine. The one lesson here is that a 24 to 48 hour culture completely rescues the phenotype. So to the NIH, I said, I have hypothesis-driven remedies to what I think ails MSC-like cells in their utility as a cell pharmaceutical for immune disorders. To the problem of donor variants and immunogenicity, one may consider using autologous cell when feasible. Don't forget that those 4,753 mouse papers, most of the times the MSCs used in the mouse model are syngeneic. Black 6 to black 6, BOB-C to BOB-C. So that's like autologous. Culture expansion, I'd advocate low passage, European style. No more than 5 to 10 doses. I understand, you want to make 10,000 doses, I get it, but there's reasons why that may not work. And I think the money shot is really to avoid the freezer burn. That's the low-hanging fruit. So this informed the clinical trial that we initiated at Emory using autologous, marrow-derived MSCs, fresh, for Crohn's disease. And we did a dose escalation, a single dose, 2 million, 5 million, 10 million cells per kilo. It's well tolerated. We got a nice signal. We only could give one dose because the FDA was so impressed that fresh cells was different from everything else. They thought it was an entire new cellular pharmaceutical, and they did not allow us for our first in-human study to do multi-dosing, which will follow. The data from this trial will be presented at the ISCT in Singapore in May 2016. I invite you to go. The clinical PI was Subra and Tanvi Derry, and I think KY is in the audience here somewhere. Thank you, KY, for all those bone marrow aspirates. This has also informed a clinical trial led by Muna Kayed and Ned Waller, where here we're using autologous marrow-derived stromal cells from patients that have established graft-versus-host disease. We collect the marrow after they have GVH and using autologous marrow to treat that. We've now have 3 patients treated. So it's feasible to use autologous cells, fresh, and we're basically asking a scientific question. Because penultimately, when we do a study where the primary endpoint is efficacy, if we do achieve a substantial endpoint result, then if you see the difference, then you can go back to a thawed product and try to reverse engineer it to achieve the same endpoints. So what's the future of the field? I propose to you MSC version 2.0. I think that the universal donor MSC platform has a role to play, and that's for the acute injury syndromes, where autologous cells is simply not practical or feasible. If you have a stroke, a heart attack, acute respiratory distress syndrome, radiation exposure, there's no time for 2 to 3 weeks to grow your cells. It has to be off the shelf. And alloimmunization doesn't matter because it takes 2 to 3 weeks to get alloimmunized. And these diseases, the outcome plays itself out in the first week. But I think you need to resolve the riddle of post-thaw cell injury. And the other thing is, and that's now a growing field, is the notion of augmenting the MSC functionality prior to transfusion. I told you that interferon gamma augments the activity, and we're sort of kind of dependent for the MSCs to get activated sort of kind of on their own in vivo. Why not pre-activate them in a Petri dish in a controlled manner? You could have an enhanced pharmaceutical. I'm talking about interferon gamma. It could be other means of augmenting the cells all the way up to gene enhancement. And of course, decipher the mechanism of action of in vivo. So I think for acute injury syndromes where time is of the essence and you need to act within 3 weeks and no long-term use is anticipated, that fresh, it could be culture rescued, but at least fit allogeneic cells is the way to go, and maybe even pre-activated allogeneic cells. For chronic inflammatory immune disorders, I'm talking about MS, Crohn's, those ailments that you anticipate that you're going to be giving this regularly, probably the optimal platform to bias it in your favor would be autologous MSCs. And I'm unbiased. Adipose or bone marrow derived, I think it's sort of the same difference, honestly. Which brings me, I know that it's very industry driven, so I'm going to make a few points about cell therapy industry and the sustainable pricing strategy, in particular having to do with the MSC world in particular. Everybody knows about Dendreon. It was the first FDA-approved cell-based vaccine technology in the US. And they went bankrupt, was it this year or last year? So they won the battle of FDA marketing approval, yet they lost the war of business and sustainability. And why is that so? It's a combination of two things, price and substantial effect. And now, people at Prochymal, from Osiris, which has now been acquired by Mesoblast, by the way, I have no conflict of interest. I have nothing to do with either outfit. They got partial approval for use of the Prochymal product, that's the Osiris MSCs, for pediatric graft-versus-host disease in Canada and New Zealand. So I'm Canadian, I'm a hematologist. I go back once in a while. I'm from Montreal, McGill. The largest pediatric BMT center in the eastern Canada is in Montreal, St. Justin's Hospital. I go see my friends, and I ask him, since Prochymal has been approved in Canada for pediatric GVH, how many of the children with GVH in your practice here, the largest eastern Canada operation, have been treated? So how many have been treated? Show of hands, 100 kids? 50 kids? 50? Okay, you're bold. 0. You're on, bang on. Not a single child has been treated, even though it's been approved. You're going to say, why? Why has no kid been treated? Because, and this is not my words, this is on the net. You know, you gotta love the net. The transcripts of the 2013 Q1 discussion, Osiris quoting Chuck Mills, who's now the new head of CERM, blah, 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 blah. Price decided we were looking at $200,000 for a course of therapy in the pediatric setting. So they wanted $200,000 for a course of therapy in the pediatric setting of their product. I can tell you in Canada, that's a non-starter. Especially if you have a product that's struggling to demonstrate that it's better than a sugar pill. So I would say, I think a word of caution and realism is if you're a startup and you have your business model and it's $500,000 per course of treatment. By the way, the $500,000 is what Novartis would like to charge if their CAR T-cell product gets marketing approval. But if you're going to charge that, you have to get a CAR T effect. A Lazarus effect. You have to cure death, pretty much. And the reason the Dendreon product failed, they were asking $93,000 for a course of treatment for a product for men with prostate cancer, and the survival advantage on the average was 4.1 months. So it was statistically significant. That's why they got FDA marketing approval. So a word of warning is, I think if you're going to move forward with the platform, I think you have to move forward with your best foot. If you're going to say, well, you know what, a thawed allogeneic product expanded for eons, it might be good enough just to make it. Even if you do that, may not be enough for you to win the war at the end of the day. So I think there's a role for academic health centers. I'm an academician at Emory, and I think the role of academic health centers is that we need to be thought leaders because we can say whatever the heck we want in front of audiences. And I think we need to innovate in sustainable. I think that's the key. You know, if it's $200,000 and nobody uses it, it's a non-starter. And high evidence-based medicine cell therapy in the academic health center space. And I think we have a role to play to de-risk industrial deployment. And that's what we're doing at our center, the Emory Personalized Immunotherapy Center. We're helping startups who don't have all the infrastructure to engage in first-in-human studies, and we're motivated. We think cell therapy is actually a very interesting cell-based platform. I haven't touched a pipette man in years. This is the team at Emory. Raghavan did a lot of the work. Ian Copeland, a good friend of mine, another Canadian that I brought south with me when I came here 6 years ago, tragically died prematurely at the young age of 42 this summer, so this talk today is dedicated to his memory. And these are the sponsors of much of the data presented today. And for those who are interested, the MSC, the premier MSC meeting is the International Society of Cell Therapy, next meeting in Singapore in May. So if you're interested, there you go. Thank you much for your attention.